So you want to know more about isotopes and how and why we measure water-stable isotopes in soil and tree water directly in the field? Let's start with the basics. Plant water uptake is essential in terrestrial water cycling. Especially trees with their extensive root systems can return a lot of water from soils to the atmosphere, also from far below the surface. It is not only important how much water they take up underground, but also where they get this water from, because this impacts water availability underground and, for example, also affects how much water is recharged to groundwater. Also, trees with a shallower root system are likely more vulnerable to increasing and intensifying droughts in the future. Isotopes are atoms of the same element, but with different amount of nutrients. Kind of like different versions of an element, but with slightly different masses. Water-stable isotopes have been used for decades to figure out where trees get their water from. That's because water sources naturally often differ in their isotopic composition, because of distinct inputs from precipitation and other physical fractionation processes, for example, isotopic enrichment during soil evaporation. And the isotope values of tree xylem water reflect the mixture of water taken up underground. Natural differences can be enhanced or altered by adding labeled water usually enriched in heavy hydrogen isotopes. Water-stable isotopes have been used for decades to investigate root water uptake. However, for a long time, samples had to be collected destructively and water was extracted in the lab and measured afterwards. This limited the temporal resolution of observations. However, root water uptake is a complex and highly dynamic process and could just not be represented sufficiently with those established methods. This all changed when new devices for measuring water-stable isotopes were introduced around 15 years ago. They were cheaper, more robust, and could be taken to the field directly. With this development, new methods were invented and applied to measure water-stable isotopes in soil and tree xylem water directly in the field, or in situ, as we also say. All those in situ methods follow a similar principle. At the measurement point, so either a certain soil depth or within tree xylem, water molecules are taken up by an airstream. The ratio of different isotopes in the sampled, water-saturated air is in isotopic equilibrium with the liquid water at the measurement location. By also recording the temperature, we can calculate liquid isotope values out of the measured vapor values. During our first experiment in the Costa Rican dry forest back in 2019, we measured water-stable isotopes throughout the dry season across 2 soil profiles and in 3 tree individuals for each of the 2 species, Caoba and Tempiscal. For each of those 6 measured individuals, we installed 3 so-called boreholes to measure water-stable isotopes in the xylem of the tree's trunk and 2 big roots. With a custom-made system consisting of pumps, electronic valves, mass flow controllers, dry air, and many connections, water vapor from more than 30 different sources or measurement points in tree xylem and different soil depth was analyzed in real time. Quite cool, though. We observed how water-stable isotopes within the trees changed when we irrigated the trees with water enriched in heavy hydrogen isotopes and with the onset of the rainy season. If you want to know more about what we found, you can have a close look at the data we collected later in this tour.